time. So I know that there's a soil seminar at 4 and that some folks want to get to. So thank you all for coming to our second Iowa Nutrient Research Center seminar. If those of you, well, either whether you are here or not, the previous seminar is up on the INRC website, Dr. Pete Nowak's seminar there, so you can go back, and the audio turned out pretty good on that. So I think that you know, worked fairly well how we have this situated. So it's my real pleasure to introduce Dr. Dan Janes, a soil scientist, recently retired from the USDA Agricultural Research Service National Laboratory for Agriculture and the Environment, has done probably as much work in monitoring nitrate export from agricultural lands in the Midwest as anybody else. And so I think it's a unique opportunity to hear about some of the work that he has done, and I'll leave him to describe his title, because Dan got creative with his title, which is great. So I'll turn it over to Dr. Janes. Let me make sure that
this is not working real good.
I'm going to turn it on. Okay.
Good. And yeah, thank you for all showing up. It's a bigger crowd than I was expecting, actually. And it's good to see all the Tilt Lab people coming all the way across campus to probably get out of doing work and listening to me today. But yeah, this is the series here, and you probably looked on the program and you saw that what Matt put down as a title is the history of water quality research at NLA, and I certainly can't cover that very extensive list of research that's been going on in water quality. So what I'm going to do instead, more appropriately, the title would be NO3 and Me, my adventures and misadventures with nitrate and agriculture. So I'm going to kind of give you a brief history of the last 30 years of my research since I came to Iowa State. And I did come here in 1990, and part of 1990, actually the money that really paid for my position was part of the MSEA, or Management System Evaluation Area, which was a program that started by President George Bush I. And he was trying to get the different federal agencies together to look at water quality, and specifically, then it was looking at herbicides, pesticides in water. That was the real issue right then. But we were also doing a little bit of work with nutrients. And one of the things that we did was to go into a watershed situation. Here I show the map here for Walnut Creek, which is a small 12,000 acre watershed just south of town. You can see Ames up there at the top border there. And do some water monitoring out there to see just what is the impact of agricultural practices on the water quality in that stream. And we have a lot of the people in here today that actually go out there and take those water samples and that, so I'm glad to see they're here too. But that was the first thing that we did. And of course, we went out there, and I'm just showing you the first 6 years of data here. But we measured flow, obviously, from many different points out in the watershed. We were looking at herbicides, so atrazine and metallic chlor, their concentrations in that water was also very important for us. But one of the other things that we also did was look at nitrate out there in that watershed. And this is actually the mouth of the watershed, looking at what's going on out there throughout those years. And the one thing that we found from this was that really the loads of pesticides really weren't that bad, at least the ones we were measuring. They rarely exceeded the MCL, or maximum contaminant level, for those products in water. But if you looked at the nitrate, what we found was that, well, that almost always exceeded the MCL for nitrate, which is, the 10 ppm level. And so really what the problem we have out there as far as agricultural practices affecting water quality was on nitrate, not on the pesticides that they were using out in that watershed. So this is also the time when there's a lot more interest now in nitrate and phosphorus in our surface waters. You see a lot of news clips coming out of the Des Moines Register. And this is also the time when the hypoxic problem down in the Gulf was starting to pick up some momentum and some interest, especially in the early 90s there. And so there's getting to be a lot of interest and concern about how much nitrate was coming out of Iowa soils and going down to the Mississippi River down to the Gulf. So knowing that the Walnut Creek, one of the big issues we had there was with nitrate, the question we asked ourselves was, well, what can we do to study the impacts of some sort of BMP or best management practice on reducing those nitrates. And this again back is 1995, 1996. There wasn't much going on during that time as far as looking at, you know, fertilizer practices, et cetera, to reduce water or improve water quality. And so the one thing that we had here was in Walnut Creek is that there was actually 3 sub watersheds in there, the 210, 220, 230. And these are basically county district drains that outlet that we were monitoring over time. And so what we wanted to do is take a watershed approach to this. We could look at modifying practices in one of the watersheds and compare it with water quality in the other 2. The one we picked for modification was the one in the middle there, the 221, which was composed of 16 fields farmed by 8 different farmers, and they were always in a corn soybean rotation then. And that's what's kind of shown in insert up there. But the question was, well, what can we do in that watershed to actually impact the nitrate coming out of the district drain there at the bottom of 220? And the thing that we came up with was the late spring nitrogen test. This is something that Fred Blackmer here in the agronomy department had been working on, and previously the last few years before this, and it looked like an approach that we could use. And essentially what it would talk about was, well, you apply a nominal amount of nitrogen right at pre-plant or just before planting. You then come back at that 6 to 12-inch stage in the corn growth and take soil samples. And then there's a formula that he had worked out to come up with, well, how many pounds per acre of fertilizer does that soil then need to bring full crop or full corn yield out of that location. And then so you come back and side dress that again, you know, right as quickly as you can. So your plants are still really small, and you can get through there with you know, a side dress application for that nitrogen. And so that was the approach that we used. We worked with the 8 different farmers that were in that location. They did everything like they normally did, but we managed their nitrogen for them. And we went out there and we would take these soil samples from every one of the fields, and then we'd just give an average application for that field based on the average recommendation from the LSNT. Now I won't go through all the nitty gritty of how we did this all, but just looking at the results here for the 4 years that we ran this program. And this is the fertilizer that was applied. The blue ones are what the farmers were doing in fields that they weren't outside of that watershed. And the red one comes up with the LSNT. And it might surprise some people that actually in that first year, 97, the LSNT actually recommended more nitrogen than what the farmers were putting out there. But then you can see the other 3 years is that the LSNT recommended less, and so we were putting on less during those 4 years. As far as yield goes, and that's certainly what the farmers were interested in, and this is 100% is what for the high-end treatment strips out there. And you can see that the LSNT then, we were basically no different than their high rates except for 1983, where we did have a reduction in yield for 92% of what that high-end application would have given us. So this kind of illustrates the fact that we're doing pretty good. But again, there is some additional risk for the farmer, because at least 1 year out of the 4, using that approach, we cost them yield out there in that watershed. As far as water quality goes, which is what we were really looking at here now, is if you come in there and manage your nitrogen, what will the impact be on water quality? This graph here represents the nitrate concentration coming out of that watershed, starting in 93 through 2004. We're doing the difference here between the treated watershed, the LSNT watershed minus the control. One of the other 2 watersheds that we weren't touching, but the farmers were just farming the way they normally did. And so 0 would be there's no difference between those 2. And you see, in the years before we started the experiment, there really was no difference between water quality coming out of those watersheds. But then we started in 1997, we started using the LSNT out there. And what we saw during that time is it took about, I don't know, 1.5 years, 2 years before we saw any impact from what we were doing out there as far as applying the LSNT. But once that nitrate got through the soil, got into the tiles, what we were seeing there at the end of the watershed was that we were having an impact on the water quality in that watershed. And in fact, by the end of that 4 years, we were down to 8 milligrams per liter less coming out of that watershed than was coming out of the neighboring watershed. So So we said that's great. We can actually do this at the watershed scale. There might be some impacts to the farmer as far as yield goes, but yeah, it works great. We ran out of funding in 2000, and so that was the last year we did anything out there. We kept monitoring. We've been monitoring ever since then, but we weren't doing anything with the LSNT. Farmers went back to doing what they were doing, and so what did we see after that? Well, the concentration went right back because the farmers were doing the same thing they were doing before we got in there. And why was that? Well, I mean, they saw the results that we got because we shared that with them every year. But it's pretty obvious why they weren't doing it, because unlike Kent here, they didn't want to go out there and take those soil samples every year at the six-inch stage, and then have to get that processed and then get out there and side dress that within a week or two. And so that was a real limitation to using a soil-based approach to coming up with a fertilizer rate recommendation for the side dress application. Of course, there's ways of getting around that. Most obvious one would be crop sensing. We're also looking at computer models to be able to do that, which is showing some promise. But crop sensing, certainly you can do it with the SPAD meter, which is that top left one, which is basically looking at chlorophyll content, or you can using it in these proximity sensors here, which are very close to the field as you go across with your applicator, or you can use remote sensing and come through there, take your picture, look at the difference in greenness there, and then come up with some sort of prescription of what you should be doing out there as well. And this is it looks like a promising approach, because now you're letting the crop tell you is it deficient in nitrogen or not, and you don't have to go out there and do all that soil sampling, right? But there are some problems with using this crop sensing to do your variable rate. And that's illustrated here, where we're looking at SPAD meter results from a couple of different field years. And the triangles there are, we put 50 pounds of nitrogen on pre-plant, and the open circles are when we put no nitrogen out in these fields to start with. And you can see, looking at, this is a ratio of the relative chlorophyll in your test field versus what a what the SPAD meter reading would be on a well fertilizer field, and that's that 100% line. And you would expect then is if we're not putting any fertilizer on, or just putting on 50, that would start going down with time, because that field should be deficient in nitrogen over time, and that's what you're seeing here. The problem is that you see that if you put no nitrogen on, we cross what is normally thought as a sufficient level, 95%. We pass under that, somewhere around that V6 VT or V7 time in the top site. The bottom site, that happens here at V6 as well. But if you put a little fertilizer out there to start with, what happens is you delay that transition from what we think is sufficiently fertilized to deficient. And the first one, you can see that's being delayed to V10, V11. Second time to V10 as well. So now, instead of like we were doing before, putting on our side dress early at like the V6 period when the plants are pretty small, you're at V10, now the plants are this high, and if you don't get out there pretty quick, they're going to be this high. And so that's a big difference if you're trying to use the crop sensing to do it, because now your window is going to be much tighter between when you detect a difference or a deficiency and when you can actually put it on. So the question for us was, well, okay, that's true, but can we still get out there and apply it at, say, V10 or later, and get a benefit from that nitrogen? So that's what we did. We went out there at essentially B16 or so. So that's right before we're going into the reproductive stage. We would do the sensing on, we didn't do any sensing in this case. Well, we did sensing, but we're driving it basically on, we're putting on half at pre-plant and then putting half again here at the side dress. In fact, this is shown right here, where our high treatment, we're putting on almost 200 pounds per acre at emergence. The medium treatment, we're putting on about 138 kilograms per hectare. Again, at emergence, our low one, we're putting on half of that, was only 69. And our split, we're putting that 69 at emergence, and then another 69 at this mid-season or V16 period. And the idea is, well, how do these compare as far as yields and water quality go? This slide here then represents the yield and the water quality all in one slide. The height of the columns there represents the yield coming out of the different treatments, which are listed on the bottom. And on the right side there is the concentration of nitrate coming out of the tiles on average for that year. And those are the lines and the dots here, or squares here. And we have 2 years of corn production and 2 years of soybean. Of course, we're not putting anything out in soybean years, but we put those in because you still get some water quality signals coming out of those years. And just to see if the nitrate practices that we're doing is affecting soybean yields at all. The answer to that one is no. Soybean doesn't really care what happened the year before as far as your nitrate applications. There's no difference across the different treatments with that. You can see with the different nitrate rates here, though, the low rate that definitely comes in lower both years. The medium and high rates were the same in 1 year, a little bit different in the second year. But when we look at that late side dress, the mid-season side dress, we see that even though we put as much nitrogen total here on this one there as we did on this middle one here, the middle medium rate, we don't yield as well. We're essentially getting the nitrate out there, but it's a little too late. The crop can't take all of it or can't take it fast enough. We've already seen, because a lot of times they're showing deficiency according to the SPAD meter, they're already suffering a little bit before we put that side dress on. And so it's really not a good practice as far as yield goes. And you can see from water quality too, that this rate here, our nitrate concentration coming out of that side dress is higher than what's coming out of the same rate, but I'll put on at emergence. So, and that's true for all the years here. So looks like you can do it. You can save some yield, but you're not as efficient as it would be if you put all that, the same amount of nitrogen down at emergence rather than trying to do a split and doing it late in the year, doing that mid-season period. So it does look to us that it's not a good idea to try to use plant sensing just because the plant does not tell us its deficiency until too late in the season under our kind of rain-fed agricultural conditions. Works better out in Nebraska where you're irrigating because you can always throw a little bit of nitrogen into that irrigation water and get it out there pretty quick. But it's really tough to do that here when your plants are starting to get up above 4 or 5 feet. That's not to say, again, that we're pretty, I'm pretty hot on side dressing. I think side dressing is a good way of doing it because when you look at when the crop is really starting to grow during this period here, and that's where that real nitrate demand is, you can get your nitrate down at that time, your nitrogen down at that time, there's less likely, it's less likely to be lost through leaching or denitrification or ammonification, whatever you want to look at, and a better chance of getting it into the crop. So we are still pretty, I'm pretty high on that idea. The problem that you get, again, is that comparing everything to a pre-plant yield-wise, the B6 looks like, yeah, maybe you get a little yield benefit out of that overall. Gets up to about B10, and B10 is getting to be plants about this high or so, it's about a wash. It's not really helping us if you're just putting the same amount of rate down. Again, the whole idea of doing side dress is that you can use something like soil testing or plants or anything to tell you how much nitrogen you should be side dressing. If you're just putting down the same rate and just looking at timing, there's not a huge benefit there. A little bit of B6, nothing at B10. If you wait all the way to B16, you're going to be losing yield out there. And actually, you're going to have worse water quality too if you wait that long. But it's still, I think, a valid approach to use. But it works best if you can also adjust the rate based on what the need is, and you need some indicator of what that need is. Reduced fertilizer rates. Again, this is, you've seen all this, yield response curve. Again, we're putting on low, medium, and high, and they're about the same as what they were before. This is a different study. And the columns, again, represent crop yield, and the lines represent nitrate concentration coming out of those tiles over that year. And what you're seeing is kind of that response function that we normally see is that the yields will kind of plateau. Here we saw a little bit of benefit of that high rate. Here, nothing. But the nitrate concentrations keep going up as you put more and more nitrogen onto those crops. So there's been suggestions, well, what we should be doing is just putting on less nitrogen. And we could, because of the steepness of this curve here, as the nitrate loss curve, putting on less nitrogen, we could get down on this end of that loss curve. The problem with that is shown in this graph here, where again, we're looking at the 60 pounds, 120 pounds, 180 pounds, and we're doing a partial nitrogen balance for what we're doing over the, this is a 10-year period that we've got here. And so for as far as inputs, we're looking at, we know we're putting in nitrogen with fertilizer. We know some comes in with the rain. There's some in dry deposition out of the atmosphere. And of course, the soybeans, this is the corn soybean rotation, will be fixing some. As far as output, you can see, well, we're taking nitrogen off in the grain of corn and the grain of soybeans. We're also getting nitrate loss through the tiles themselves during the corn year and the bean year. And there's probably also denitrification going on out in those fields. And so we can sum those up for these different studies. And this is what we get as far as the nitrogen balance is. And you can see that we go up to the high rate there of 180 pounds per acre. We got just about comparable numbers there. Really within our errors, those are the same numbers. So we're taking off as much nitrogen as we're putting into the system. We go down to this low one, we're taking off a lot more nitrogen than what we're putting into the system. And then the question would be, well, where's that 240 kilograms per hectare coming for? That's not accounted, that's the mismatch between those two. And the only place left that it could be coming from is from the soil itself. Soil mineralization, we're basically mining soil carbon out of that system. It's mineralizing, we're getting nitrogen out of that, and that's what's going into corn. And so if we do this continually for a long time, it could be an issue. And even this medium rate, which was close to what the optimum economic rate should be for that field, we're still seeing that there's a mismatch there. We're taking more nitrogen off the field than what's going into the field. And if you look at that over 10 years, that represents a 2% loss of either soil organic nitrogen, soil organic carbon. So we're mining this system. And of course, that's not a sustainable practice, because we want to keep our soil organic carbon at its current state, or even bring it up in a lot of fields. And in this case, we're mining some of that out of there. And so that's certainly not sustainable in this case. And that's something we have to worry about. Anything that we develop as far as these practices go is, are we sustainable as far as soil organic matter goes? Looking at some other studies that are out there, and also saw a soil end change here. You can see on the right side there. There's only 3 here that really had a positive impact. And those essentially had either a lot of nitrogen put down in a corn soybean rotation, or a lot of nitrogen plus a cover crop, which is putting more biomass, more carbon into that system, or you're putting down a lot of swine manure as well. Those are the only systems that we saw in the literature that were getting a positive response as far as soil organic carbon goes. So it is an issue that we need to worry about. Another thing was being tossed around a lot in the late 90s was precision ag. And it's still being talked about today. We'll talk a little bit about what we looked at there. And we know, especially when the yield monitors became a real hot item, and every farmer had one, that yields vary across the field. Now, we've known that forever, but it became real obvious when we had a yield monitor out there. You got a map back to something like this. And you said, well, geez, what's that variation out there, and what can I do about it? And as a soil scientist, of course, I looked at that as well as a lot of other soil scientists and said, well, it must depend on the soil and landscape properties out there. That's why we're seeing those differences. And since, you know, I'm a smart soils guy, I can probably identify what the problem is out there, and we could fix it based on just our knowledge of the soil properties across that field. So that's what we looked at. That's what we thought, well, that sounds like a good idea. Let's see if that pans out. So we went out to a field of, this is in Boone County. There's 224 microplots and 8 transects here. And you can see them there. And what we did is, we didn't do it, we just let the farmer do his job out there. But what we did was come in there and harvest each one of those strips before he came out and harvested the field. And so we'd harvest, I forget what those lengths were, 40 feet or something like that, with a small combine, let the thing clean out so we know exactly how much yield came out of that one little square, and we move on to the next one. We do it all the way across the field then. And so we could build up a very precise a measure of what the yield variability was based on those 224 locations. And this is what it kind of looked like. This is the 1999 results here, and you can see the different yields going across that field. And this is not yield monitor data. This is precise basically way wagon data. So this is real clean data as far as the precision of what we're seeing there. And we can do this every year. So once we got that, then we come back. Well, okay, so we're assuming soil has can explain what's going on out there. And so we also went out there and took a lot of different soils and landscape measurements. This is a list here of the things that we measured at each one of those 224 locations. And we said, well, we're hoping that these things here, what we suspect to have an impact on yield. And so there should be a relationship between these soil properties and landscape properties and yield. And so Tom Casper, who is one of my colleagues, now retired at the TILF lab, he did a factor analysis looking at those, basing those on 6 years of corn data that we had out of that field. And then this table here just shows factor analysis that he did on that. We have 4 factors that were important. The first factor you can see on with the little footnotes there that what was important in that factor, and there were things like soil texture, carbon, soil color. They all help contribute to that factor 1. When you got down to factor 4, the only thing that was really contributing significantly to that was profile curvature in the landscape. But based on those then, those 4 factors, we could predict the yield of corn and soybean across over those 6 years for that field. The only problem that we had was, and you can see we had a pretty high R squared as far as what the factor analysis is doing and what the yield was. The only problem was then, though, the only way we could do that was you had to split the dry years, both corn or soybean, from the wet years. So although we can predict the yield, we have to know, is it a wet year or is it a dry year? Of course, you don't know that till the end of the year, so it's not much prediction there, right? If you can't predict it before you start growing your corn or soybeans, it's not going to do you much good. So that was a frustration, is that, yeah, we can relate this back to soil properties, but it really depends on the weather too, as to how precipitation especially interacts with those soil properties to give you plant yield. And of course, that's not surprising to anyone. If you look at yield from a 40-acre field over time, yields not only vary with space, they vary with time. And that's the same field for different years. It's corn on the right, corn on the left, soybean on the right, and you can see we got quite a bit of variability over time in that one field. So what we try doing instead now is forget the factor analysis and soil properties. Let's just look at the yields themselves. And we'll do a cluster analysis on those yields from those 6 years. Just the corn we're looking at right here. And essentially, if point A behaves the same over the years as point B, we're going to put those in the same response cluster. They're acting the same. And point C might act something different, and that would be in a different cluster. And so that's what we want to do as far as cluster analysis, is just look at those that are behaving kind of similarly over time. And this is the results that we found, again, represented on those 224 locations, is that we were able to cluster them down into 5 different behavior classes. And as you see there. And again, these are different, these are clusters that are behaving similarly over time. And so the clusters were also spatially contiguous, which is not part of the cluster analysis itself. It's not a spatial analysis. The fact that they're actually clustering together is gratifying, because you'd expect that soils next to each other behave more similarly than soils that are farther away. They're also analogous to landscape positions. The one cluster we have in the middle there, the blue one, was actually at the middle of a small pothole that was out in that field. And some of the yellow ones were ones that were kind of up on summit locations. And ones in between were in different location, landscape positions as well. And the other thing was that the yields in clusters were influenced by growing season precipitation. And that can be seen here, where we're looking at those 6 years, mapping the different clusters here, how they behaved each one of the years. And so if you take something like the pothole, the yellow ones there, and the pothole is just kind of a name that we gave it. But you can see in the wet years, as you might expect, well, it just floods out. We don't get any yield out of there. But in a dry year, the pothole is going to be the best yielding location out in that field. And so now we've got 5 locations, and you can look at, like, say, the summit location here. it's not really impacted much at all by precipitation, but it's almost always a low yielding a cluster. So we got 5 clusters out there. We ended up having to combine the toe slope and the pothole because there wasn't enough spatial extent of either one of them for us to work with. We just combined those into one. Then we can come back and say, using multiple discriminant analysis, say, all right, we know what these are. Can we relate those to some easily measured soil and landscape properties? I list the ones that we looked at here. And it turns out that, yes, you can actually map those yield clusters based on elevation. You can see the list there. And so what you come up with now is using that information, and because I can do elevations, I can do all that based on an elevation map or a LiDAR map. Soils color you get, again, from an air photo. So I can interpolate between all those 224 locations I had, and I can come up with this map now. This is the extent of those different yield zones across that 40-acre field. So that's pretty exciting. It was fairly successful. We accurately predicted about 80% of the cluster identifications here. The nice thing about this now is we can go into, say, the north 40 on that field, and I can also predict those yield zones without having any yield data now, which is typically what you do when you go into a farmer's field. They don't have a good map, well, they didn't back then have a good idea of what their yield variability was over time. And the other nice thing, even better, is we could go into a neighboring field, one that's still in that Clara Nicolette Webster kind of soil sequence. And this one was 10 miles away, and we can still predict what those where those yield clusters are across that field. So that's kind of nice. Now we have a tool that can actually predict those yield zones in any field in this, say, around the Ames area. That still begs the question, all right, we know the yield zones, we can map out yield zones, but can we manage N based on those yield zones or not? We don't know that at this point. And so what we had to do then is go into these fields with the map of the yield zones and overlay a nitrogen rate response trials on top of those, where we went from 0 to 200 pounds per acre in 40-pound increments. And you can see these are all represented within, say, this is the pothole area here. And then we did this in triplicate across not only this field, but several other fields as well, to see what the yield response was within that zone compared to the other zones. And what we got is shown here. This is 6 years of different field plots or field years here. And indeed, there is a difference in the nitrogen yield response based on those different yield zones that we identified out there. And that was very gratifying. Then you come back, and here's shown in 3 years where we can actually measure, or afterwards, calculate what the economic optimum nitrogen rate was for those different zones for those different years. And you can see in 2001 that, yeah, for those 4 different yield zones, there's definitely a difference between the EONR for those 4. The problem you get is, like in 2003, you can see the blue one is the pothole one, and the EONR somewhere around 225 pounds per acre, but in 2005, the EONR is 0. So they're still bouncing around depending on, again, precipitation mostly. And so, yeah, they are uniform, but they still depend on weather. So it's still pretty frustrating as far as that goes, as far as how do you manage nitrogen, because, you know, before the season, you don't know if it's going to be a wet year or a dry year. But you can still look at the average EONR for those different zones and see, is that still a way of approaching maximizing profitability within that soil or within that field. And so we looked at that with a certain nitrogen cost and a certain yield or bushel per acre price for corn. And this is the results that we got for those 6 years, looking at the difference between if you do it by zone or if you do it just a single rate across the field, which is what the farmer would be doing normally. And you can see that there are some years where we came up negative, that the farmer was making more money just by treating the whole field as one zone versus using it like we would have it as 4 different zones. But overall, on the average, the farmer would actually be saving $9 per hectare by treating the four different zones rather than treating it as a uniform field. So it still has some promise here based on those yield zones. And so I think that's a very fruitful way of maybe looking at trying to improve your nitrogen rate across the landscape. And this kind of review again, we found basically that our zones were kind of similar to what the landscape positions are. So if you look at the maximum yield across those averaged over time, you see that, yeah, the potholes the toe slope pothole area had the greatest yields, about 13 megagrams per hectare. And as you go up slope, it decreases until you get up to the summit here where you get down to 10. And that makes sense because the pothole is very high in soil organic carbon. It's getting new deposition of carbon from erosion off other locations every few years, whereas the top there, the summit there, is eroding. It's got a very thin soil organic or A horizon to it, and we're just not getting the yields out of there. So this all kind of makes sense. I'm sure this is what you would expect to see out there in the field. What's perhaps a little more surprising is if you look at now what is the economic optimum rate for those different locations, what you see is that actually the lowest economic rate is down here in the pothole. It's only 128 kilograms per hectare. The highest one is up here at the top, 219. And I know I've worked with a lot of crop consultants, et cetera, that have thought just the opposite, that, okay, well, it's a high yielding location. We got to put a lot of nitrogen on there because it's taking a lot of nitrogen off. And so they put more of the nitrogen down at the bottom and put less up on top. But that would not be the way to maximize the return nitrogen applied to that farmer. Instead, it would be just the opposite. And why was that? Explanation is pretty simple, is that there's an extra nitrogen coming out of that system basically from mineralization from the soil. And so again, we did a soils budget on that. What we saw is that when you're up there on that eroded area, you're not getting much nitrogen coming out of the soil. You're down in a pothole, you're getting a ton of nitrogen coming out, mineralizing out of that soil. And so you don't need to put a lot of fertilizer down in that location. So it's just a nice way of representing the fact that, the yields might kind of go like we would think, but when we talk about, well, what do you do with that? How do you manage your nitrogen? It may be opposite of how you would actually approach it, and what a lot of people have done in the past, and probably are still doing, as far as how do you manage your nitrogen across the landscape like that. I also worked with fall cover crops. Tom Casper from our lab been working with cover crops for about 30 years. He was working on them before I even came to the lab. But what we were interested in is what's the impact of water quality from fall cover crops, particularly for this is rye being planted in the fall. The idea is pretty simple there, is that typical curve here over the year, where we're looking at precipitation, ET from the crop, N uptake, which also depends on ET, and tile drainage, which is happening in the spring and a little bit in the fall. But what we like to do, it's a fairly short season when we're actually having something evaporating, transpiring, and taking up nitrogen, a growing plant, that is. What we like to do is kind of spread that out, get that curve widen it out. You're not going to be able to do that with corn and soybean, but if we plant a cover crop in the fall that can start growing in the fall and continue growing in the spring, we could effectively get more ET, more nitrogen uptake, and that would solve some of our problems. So we started working on that in 2000, I guess. This is the results from 2001. So this is actually the first year that we had drainage when we were starting our comparison. And this just shows the results of the blue is having no cover crop. This is no-till ground. And the pink there was the cover crop. In 2001, they're both pretty much the same at the end of the drainage season, which was looks like at the end of July or something. Things stopped flowing. We then put in our cover crop in the fall. It was rye, so it was growing that spring. It continued growing that fall. It continued growing that spring. And when the tile started flowing the very next year, we saw a huge change between what was going on with the plot without cover crops and what was going on with the plot with cover crops. In fact, I don't really have a good explanation as to why we should see such a huge difference just over one season, basically over fall, winter, spring. But that's what we saw in the numbers. And in fact, over the last 18 years, this is what we've got as far as the blue, or I guess the green, or what that color is the conventional drainage, and the blue represents the cover crop, the rye cover crop on the same, on adjacent plots. And you see all except for 2001 that the cover crop was showing a lot less nitrate coming out of the tiles. Here I'm just showing you concentration, but it'd be the same for load. And in fact, on average, you can see down here over the 18 years, we got about a 52% reduction in nitrate coming out of the tiles when we're putting on annually, we're putting on a rye cover crop in the fall. The other side of this, of course, what the farmers are interested in is what that cover crop doing to my yields. And that's shown here. So this is the yield here. Again, with the blue is with the cover crop and the orange or whatever, green rather, whatever that is conventional. And you see there that typically we're getting a little reduction in yield with our rye cover crop. And I'm not going to go into the reasons for that. It's been looked at quite a bit. But on average, you can see for us, we see about a 3% reduction in yield for corn following a rye cover crop. So that, you know, if you're getting 200 pounds or 200 bushels, you got 6 bushel loss in yield when you're doing cover crops. That's one reason why farmers aren't too excited about cover crops, because it could potentially cost them yield. Plus, it costs maybe $30 an acre to put the cover crop out there. And I know there's others that have had successfully been able to get even greater yields with cover crops, but on this field, we have not been successful in doing that. We've tried a lot of different, I should say, Tom Castor's tried a lot of different things. So one way of getting around this is, well, don't plant rye, right? We don't have to use rye as a cover crop. One thing we use is oats. And the nice thing about oat is it grows a lot more in the fall, and then it dies over the winter, and it won't be growing in the spring. And so you don't get a lot of that interaction between the cover crop and the next year's corn. And so this shows you 5 years here where the corn years would be the even years. The odd years here were soybean years. But you can see as far as the nitrate concentrations coming out of the tiles, again, with the conventional here, here's our rye one, and our oat is not as effective because it's not growing in the spring, but it was in the fall. But it does something, and so you do have a reduction every year, but maybe not as good as if you had rye that's getting you more growth, or giving you some growth in the spring compared to the oat that's not giving you anything. So oat would be a pretty good alternative. We don't suspect, I can't remember what the data showed, any yield drag with the oat or corn planted after oat. But it's not, you won't get quite as big of a water quality impact from it either. I thought I took this one out, but I'll skip it. Tile drainage design then. So we did a lot of stuff in the field. We basically thought, well, you can kind of nibble around the edges, but if a farmer's doing a pretty good job with their nitrogen, if they're good, good managers of their nitrogen, there's not a whole lot you can do with precision ag or anything else that really improve that much. So the question was, well, what can we do besides on the edge of the field or out in the rivers? And one of these things was looking at tile drainage. You know, here in Iowa, we have a lot of tile drainage. Across the Midwest, across the Corn Belt, there's a lot of tile drainage. And we know that a lot of the nitrate that we're seeing going down to the Gulf of Mexico is coming out of those tiles. So one thing we could do maybe to reduce that nitrate would be, well, let's decrease the drainage of water coming out of those tiles. Or maybe increase denitrification, either before it gets to the tiles or when it gets to the tiles, before it gets to the stream. So again, we can remove some of that nitrogen. So over the years, I've worked on 3 different things here, showing the schematic. I won't really spend any time with this. But the first one we looked at was bioreactors. I'm sure everyone here is familiar with denitrification bioreactors. We're using wood chips because they have good permeability. Their carbon is labile enough that it's available to bacteria, but it's not so labile that you have to recharge it, you know, except for maybe every 10 to 20 years. So it's a pretty nice medium to use. What we looked at is, rather than looking at the end of the pipe bioreactor, which is, you know, fairly typical and what they have written a standard for NRCS, we wanted to look at, well, what's the impact of if we put a wall of carbon around our drainage pipe here, so that as the water moved from the field into the tile, and it had to pass through these wood chips here, and hopefully we get denitrification there, and so the nitrate wouldn't get into the drain tile. And so this is the result here of, again, 18 years corn and soybean, but the wood chip wall is always there. It treats about 80% of the field. That last 20% between the tile and the wood chip walls is untreated. But you can see what the reductions are here against, conventional versus the wood chip. They were very effective as far as moving nitrate, as effective as the putting down cover crops. Here we see on average a 61% reduction. The nice thing about this is once we paid for it back in 2000 when we installed it, we don't have to keep paying again. Whereas cover crops, you got to keep out there and seed it every year. And if the farmer doesn't seed it every year, you're not going to get any benefit from that cover crop. So this is kind of nice. It's working. We don't see any kind of diminishing effect over time. And so in this case, it's working really well for us. The other nice thing about this is that people are worried about nitrous oxide coming off these with a powerful greenhouse gas. And since we're denitrifying, can that be an issue? We saw that there is no significant difference between the wood chip over the wood chips and over the field itself. I'm going to have to hurry up a little bit here. Drainage, controlled drainage. The idea here again is that rather than having your tile outletting, you put a control structure on there. And so in the fall or the winter and spring, you could raise up the logs here. So you're basically raising the water table in your field and reducing nitrogen or reducing tile flow. During the planting and fall harvest, you could drop it all down. And so it'd be perfectly well drained and you could get across the field. And then maybe during the summer, you could actually manage that water table, hold back some water that you might need later on in the season if things turn dry. This is I was part of a Midwest project that was looking at that impact of controlled drainage across the Midwest. This is a slide that Gary Sands and Matt Helmers put together from the results of those. And they got a slope of about 2, where you're comparing the conventional versus managed N-loss or the controlled drainage. So that's about a 50% reduction that we saw in nitrate concentration using controlled drainage. The bad thing about it is that you would like, since the farmers have to spend money to put these things in, you'd like to see some sort of yield benefit. And from this study anyway, we didn't see any yield benefit. So that's a real problem because that means they're not getting any yield benefit to help them pay for the upfront cost of putting in the controlled drainage. The last thing I'll talk about is the saturated riparian buffers. This is what I've been working on the last 10 years with Tom Eisenhart here. And we're trying to take advantage of a buffer, and we'll just skip through this pretty quick. But we know that the buffers next to the stream can have a lot of mediating impact as far as removing nitrate and phosphorus, or taking up holding sediment, taking out pesticides, etc. The problem that we have here in Iowa is that since we have extensive tile drainage, we basically bypass, all the water bypasses those riparian buffers through the tile and just gets dumped out in the stream directly. And so what we wanted to do was come back in and replumb that system to see if we can't get more interaction between the tile water and the riparian buffer. And that's what's shown up in here, where essentially we put a diverter box and put in a new tile here, where we're diverting water out into that tile. The water leaks out of the tile into the soil as shallow groundwater, moves to the stream. As it does so, we're hoping that we're getting some nitrate removal. Because again, typically these buffers have a lot of carbon in them. It can act as a labile source of carbon for denitrifying bacteria. Here's just an overhead of installing one of these before the buffer was actually planted. And then this is a saturated buffer that's been in place for, I don't know, 5 years or so at one of the farmers' fields. You can see that they can look pretty nice. This slide here kind of summarizes the results from that. I know it's a little complicated, but we're looking at different fields here and 20 different site years. The red bars and the red line across there represents how much of the nitrate that was coming out of that tile we were able to divert out into the buffer. Because the buffer can't take all of it, especially during peak flows, but it can take a good proportion of it. And on average, you can see it takes about 54% on average. You can see that there's some locations like here, this Iowa 1, we're taking 90% of the nitrates being diverted out into the buffer, whereas there's others like the BZ2, we're only diverting maybe 30% on average. And that all depends on how much the size of tile that you're intercepting and how long the saturated buffer is. It's one thing to get it diverted, the other thing is we got to get it removed, and that's what the blue bars show, is how much nitrate, total nitrate coming out of that tile is being removed. And again, the average here is 45% or 46%. So that's really good. Again, you see in some years or some locations, we're getting 80, 90% removal, and others like SH1, we're down here around the 20% level. So they're not working as well. But overall, we're pretty happy with how well these things are working across the landscape. And these represent locations all across northern Iowa. So again, just in summary, side dressing, as long as it's based on some sort of soil test, etc., as far as adjusting the N rate, doing a side dress works really well. We saw about a 30% reduction in nitrate at the watershed scale. Basing it on crop sensing is probably more problematic because the sensor is not sensitive early enough in the season to tell you, okay, this plant's going to be running into problems. And so the application window is really too narrow. The yield zones idea works really well and was a good way of applying nitrogen. If you really wanted to work it well, you need to know wet years from dry years, but even on average, we could still save money for the farmer. We have to be careful in anything that we do that we're not mining carbon out of our soils, and so that's an important thing to look at, especially in a corn soybean rotation. If you're not putting on any cover crops or anything like that, not putting down manure, this is a real issue that we need to worry about because that talks about the sustainability of our corn, soybean, grain landscape farming systems. Oat and rye cover crops work really well. The rye works about twice as good as the oats, mainly because it's growing for a longer period of time. Controlled drainage also works really well. Unfortunately, you can't put those systems everywhere. There's some landscape criteria that you have to worry about there. Bioreactor walls and bioreactors themselves, again, work really well. They're kind of costly for putting in. They can run, $10,000, $13,000 to put one in. And we're not sure about the lifespan. If it's only 10 years versus maybe 20 years, that'll have to figure into what the cost per nitrogen removed will be. And that's still being studied as we start seeing some of the bioreactors that have been put in place. As they start aging, we start seeing if they need to be recharged or not, we'll get a better idea what that cost will be. Saturated riparian buffers work as well as the bioreactors. They're a lot cheaper to put in. It's nice because once you put them in, you can walk away from them. You don't have to manage them. The farmer doesn't have to manage them. They'll just keep on working. But a problem with those is that, again, you can't put them everywhere. They have certain landscape parameters that have to be met before you can put a saturated riparian buffer in there. I think in Iowa, we estimated that it might be, 14,000 miles of stream that we could put these in. And so it's not everything, but it, again, it would move us down the line of trying to reduce the nitrate coming out of the state of Iowa. And with that, I see I'm 2 minutes late. Well, of course, that clock's off too. So, all right, thank you. And if there's questions, time for questions now or after the seminar, I'd be pleased to stay around and answer.
Thanks, Dan. So hopefully folks can stay around just a few minutes to ask Dan a couple questions. So if anybody here has a question, I'll pass the mic just so the people online can hear the question.
Or not.
I'll ask the first question. In your 30-year career, what's been the most exciting finding you've had?
Actually, the most exciting thing is what Tom and I have been working on as far as saturated buffers, because that was something we took kind of right from the go and developed the process and the practice, and went out in the field and demonstrated that it worked better than we actually thought it would work, which was that doesn't happen very often. And then we've been able to get that adopted by NRCS. We've got these things around the state of Iowa. They're also installed in Minnesota, Illinois. I'm not sure where else. What's that? Ontario, yeah, there you
international.
Go. So they're international now. But that's been the most satisfying, I guess, Matt, just because it was really something that we took from, the beginning right up to a successful end. Other question?
So we like to focus on nitrogen a lot. How important is carbon in this? It seems a lot of these situations was soil carbon might have been sort of another factor in reducing nitrate. So maybe we apply more carbon or increase soil carbon in nitrogen could be less of a concern.
Well, certainly as far as denitrification is concerned, the carbon is important. You got to have labile carbon in the system for the bacteria to chew on. If you don't have that, you're going to have to supply it like we do with the bioreactors, putting in wood chips or whatever else you want to put in. So yeah, it's really important. And when you're talking about soil organic carbon, I mean, soil organic carbon and soil organic nitrogen are really the same thing, you know, two faces of the same coin. Once you're talking about if it's already weathered down to like the final part there, you're always talking about, you know, 10 to 12 ratio for carbon and nitrogen. So yeah, they're very important. Exactly.
Have you tried, or anyone else tried, to put saturated buffers along ditches?
That's a good question. We put saturated buffers along ditches. Actually, some of the ones we put them on, they've been straight and streams, so essentially they are ditches. But we're also got a new project starting out. I don't know if you're going to fund that one or not, Tom, but we got one going up in northeast Iowa where the farmer actually put a saturated buffer not against a ditch, but alongside a county tile drain. And so rather than the water going down to a stream or ditch, it's draining into that county culvert. And so it'd be interesting to see, if, other than that, the same exact system, it'd be interesting to see if that one works just as well. There's no reason why they shouldn't work out. The only thing would be, when you put in a ditch, that might not be an area that historically had drainage, and historically does not have alluvial soils. And we know we want to have a lot of carbon in our soil when we go into these things. You may not be in a situation where you have that carbon with depth in those soils. That's not to say that it won't work. It's going to take more time for them to work. I think Eisenhardt has some data that shows that when we put in that perennial vegetation around a buffer, we're putting carbon into that soil over time. That carbon over time then can help sustain denitrification. But in that case, it's going to take, you know, a decade or two before you get enough carbon in there to get substantial denitrification going on. Yeah. But it should still work. Yeah.
Thanks. Those differences in MRTN across the landscape are really striking, you know, with the lower recommendations in depressional areas versus the uplands. What do you see as the barriers for why that, you know, adopting that kind of variable rate has not become more popular? Is it sort of just a philosophical reason, or is it a, you know, technical kind of a barrier?
It's not technical, certainly, because you know, the technology is way ahead of anything we do as far as prescriptions go. Yeah. I mean, there's technology out there that can vary nitrogen rate, you know, every inch across the field. Yeah. So that's not the problem. The problem is in coming up with that nitrogen a recommendation. Yeah. You know, and the problem is, you know, I'm showing information that we did on one soil landscape. It's not been repeated on other soils and landscapes, and that's what we would really need, is more data so that we can feel better about it. Or once we get off the Clarion-Nicollet-Webster area here, now what do? Do we still see the same results with different kind of clusters that we can monitor for? And that data has not been gathered, as far as I know. Yeah.
Any other questions? Well, let's thank Dan again. And sounds like he's willing to stay around a few minutes and answer individual questions.
I'm retired. I got nowhere to go.
Okay, all right, stay around. And just a reminder, 1 month from now, not 1 month, but you know, the time in March, Dr. Tom Eisenhardt will be talking about kind of the history of buffer research at Iowa State University. So please come back. We'll be in the same room again. So thanks everybody for coming.